Hi, so the article we chose to evaluate is Meningeal Lymphatic Dysfunction Exacerbates Traumatic Brain Injury Pathogenesis, which was written by Bolt et al. So the traumatic brain injury, aka TBI, has negative implications on motor, cognitive, and sensory systems as well as mental health, thereby making a topic that is heavily studied in an attempt to discover more mechanisms and find better treatments for this type of injury. So the immune system is activated during a TBI as it works to dispose of neurotoxic molecules and repair the brain tissue that was damaged. However, prolonged immune response can cause neurological impairments due to the faulty disposal of damage-associated molecular patterns, DAMPs, abiotic organic matter, and protein aggregates. And the figure on the right just shows you some of the major causes of TBI. The cerebrospinal fluid, CSF. So CSF is a saline fluid that is made from the choroid plexus and pumped into the ventricles and the subarachnoid space of the central nervous system. So the fluid's main role is to regulate the extracellular environment of neurons, and it also provides chemical protection and physical protection of the brain. So it protects the brain by suspending it in fluid. So it pretty much acts as like a cushion. So the meningeal lymphatics. The meningeal lymphatics are lymphatic tissue found in the meninges of the brain that run parallel to the dural venous sinuses. They are responsible for draining cerebrospinal fluid containing the immune cells and excess organic matter from the central nervous system to the deep cervical lymph nodes, DCLN. Meningeal lymphatics also function to dispose of proteins such as beta amyloid plaques, extracellular tau, and alpha-synuclein that cause neurotoxic damage-associated molecular patterns, DAMPs. This prevents neurotoxic materials from causing brain damage. So the cerebrospinal fluid drains from the adjacent subarachnoid space into the meningeal lymphatic vessels, mainly from the dorsal side of the skull. So the CSF then makes its way into the deep cervical lymph nodes, DCLN, at the base of the skull. So looking at the image on the right, it shows the location of the meningeal lymphatics. The lymphatics are located right on top of the venous sinuses, running parallel to it in the dura mater of the brain. The arrow at the top shows the flow of the cerebrospinal fluid from the subarachnoid space to the meningeal lymphatics. So this article explores the association between TBI and meningeal lymphatic function. The article hypothesized that TBI results in meningeal damage. The researchers found that TBI causes meningeal lymphatic impairment, specifically a rise in the intracranial pressure ICP following a TBI, and causes lymphatic drainage defects. So the TBI causes the cerebrospinal fluid drainage defects in meningeal lymphatics. So moreover, lymphatic impairments exacerbates TBI pathogenesis. So in the article, the researchers used mice in all the experiments that they conducted. To achieve the results given in the article, experiments 1 and 2 used both female and male mice, while experiment 3 used only male mice with pre-existing lymphatic dysfunction. And TBI was inflicted using a controlled cortical impact device, an image of which is shown on the right. So in their first experiment, Bolt et al. Looked at how TBI would affect the meningeal lymphatics of mice. They anesthetized the mice with 4% isoflurane before inflicting TBI on the piriform region of the brain. In the control condition, TBI was inflicted on the posterior section of the eye, but it had missed the brain. They then used immunofluorescence to track the CSF circulation in the lymphatics by using fluorospheres bead injections into the cisterna magna. The meninges, DCLN, and brain were then surgically removed to examine the localization of the beads. They found that there was low bead drainage to the DCLN in the TBI-inflicted mice. This suggests that the meningeal drainage system was damaged by TBI, which affected its ability to drain CSF and dispose of waste. The defect in the meningeal lymphatic drainage system lasted 1 month post-injury and only started to return to pre-injury levels 2 months post-injury.
Image G compares the bead drainage to the deep cervical lymph nodes of the control mice to the mice inflicted with traumatic brain injuries. From the graph, we can see that bead flow was significantly impaired in mice inflicted with traumatic brain injuries until 2 months after the onset of the injury. Meningeal lymphatics are sensitive to changes in pressure and swelling in the limited space of the skull. Therefore, this experiment aims to explore the relationship between intracranial pressure and meningeal lymphatic function. To increase intracranial pressure in the experimental mice, left and right internal jugular veins were surgically ligated together, while the control mice underwent a sham procedure to mimic the effects of the ligation. A hole was made in the skull of anesthetized mice on their left parietal lobes. A pressure sensor catheter was inserted to record changes in intracranial pressure. Fluorescent beads were injected into the mice to evaluate the drainage of cerebrospinal fluid to the deep cervical lymph nodes. Similar to the first experiment, bead drainage to the deep cervical lymph nodes was lower in the ligated mice, implying that increased intracranial pressure impairs cerebrospinal fluid drainage function in the meningeal lymphatics. The image shows the comparison of bead drainage results of the control mice versus the experimental mice. Graphs F and G suggest that some amount of lymphatic recovery had occurred as the proportion of beads drainage into the deep cervical lymph nodes from 3 hours to 24 hours post-injury had increased. Graphs I and J support this idea. Records made 3 hours post-injury and 24 hours post-injury show a decrease in the area of bead coverage in the meninges. In both sets of graphs, specimens that underwent jugular vein ligation showed less effective bead drainage, a lower average proportion of beads in the deep cervical lymph nodes, and a smaller than average change in meninges bead coverage compared to sham specimens. The last experiment studies the effects of pre-existing lymphatic dysfunction on traumatic brain injury pathogenesis. The mice used had pre-existing lymphatic dysfunction. Traumatic brain injury was inflicted using the same method as the first experiment. The meningeal lymphatic vessels were ablated selectively using intracisterna magna injection and transcranial photoconversion of Visidyne on the mice's skulls. Ablated mice were treated with Visidyne and laser. Non-ablated mice were treated with Visidyne and no laser. Image A shows an increase in ionized calcium binding and adapter molecule 1 activity in the ablated and traumatic brain injury mice compared to the non-ablated control mice 2 weeks post-injury. This increase in IBA1 staining in the ablated plus traumatic brain injury group led the researchers to analyze the post-injury IBA1 cell complex in depth. They found that the IBA1 cells in the ablated and traumatic brain injury inflicted mice had fewer and shorter dendritic branches that were further from the soma. The dendrites also had fewer branch points and a decrease in dendritic volume as seen in graph E. Cells that underwent ablation and TBI, in orange, have much fewer intersections than any other group. This suggests that pre-existing lymphatic damage may result in increased activity and decreased development of brain macrophages, of which IBA1 plus cells are a part of. Therefore, repetitive head injuries will induce faulty drainage of macrophages, leading to severe neuroinflammation and cognitive deficits. To conclude, experimental results show that traumatic brain injury induces deficits in cerebrospinal fluid circulation due to the rise in intracranial pressure. Increased intracranial pressure causes meningeal lymphatic impairment, which impedes drainage from the lymphatics to the deep cervical lymph nodes. Pre-existing lymphatic dysfunction can raise the risk of the brain developing more severe neuroinflammation and cognitive deficits. The meningeal lymphatic system is needed for drainage of cerebrospinal fluid and antigens to the deep cervical lymph nodes, which promotes autoimmunity and activates the immune system in response to brain cancers. These discoveries provide insight on the causes of certain neurological disorders that arise from traumatic brain injury and contribute to new research for developing better traumatic brain injury treatment. Thanks for watching.